My name is Gustavo. I'm from São Paulo, and I am developing this work as a part of my PhD with Thiago Quintal and in collaboration with Paulo Guimarães. The results I'm presenting today are very preliminary, early stage, so any feedback is very welcome. You can come find me during the conference, so we can discuss some ideas. And I'll start now. So, it's no secret that biological interactions are of huge importance for evolutionary biologists. So, for example, antagonist interactions such as predation, the battery is okay, such as predation or seed predation or competition, for example. And this type of interaction has been quite well studied, and also some mutualistic interactions such as pollination and seed dispersal are also studied and very important, but they don't have that much presence on like evolutionary studies. And when they do, they usually comprise just one or a few pair of species, and majorly, basically, they are focused on co-diversification or co-speciation. But in the past few decades, there has been a lot of work being done on interaction networks, where you can study a community level, encompassing a lot of different groups. And for example, this is a frugivory network, a seed dispersal network from Pantanal, where you can see that there are different groups working as seed dispersers, such as birds primarily, but also mammals and even fishes. And this is quite interesting because it provides us with a very broad ecological perspective. So you can study a lot of different species and groups at the same time. And especially when you use information theory-based metrics, such as centrality, for example, and here's just a cartoon example of a relationship network. And this is just to illustrate how when you start to, when you choose different ways of characterizing the role of each of the components of this network plays in the network, you can find different results. For example, if you choose the closeness centrality, you get those two main nodes as the main role, the major roles in the network, and so on if you choose different metrics. But one thing that is missing in network studies are a more macroevolutionary perspective. But to our help, in the last years, a lot of methods have been developed that allow us to estimate diversification rates, such as speciation and extinction rates, with a lot of confidence. And also with the development of modern molecular phylogeny methods, we could assemble a huge data set. For example, for birds, you can see the whole phylogeny for birds, and this allows us to have a large amount of data that can be used to investigate the group. So based on those two types of data, network data and diversification data, one question came to our minds. So is the diversification dynamics somehow linked to the ecological role that species play in an interaction network? For example, some hypotheses. Here's just one of the cartoon examples. And for example, central species, the species that have an important role in this network, do they have, like, for example, lower net diversification that comes from lower extinction and lower speciation rates? Usually those rates can be linked. So if you want more detail, you can go check Thiago's presentation on Monday. And by having lower net diversification, or a slow pace of evolution, these species could be a predictable resource for plants to adapt, for example. On the other hand, peripheral species probably show a higher net diversification or a high turnover, and with that they could be less stable in deep time, and thus being less predictable and building very few interactions. So how do we plan to address this question? So first, we have here an example of a bipartite network on the top row we have the birds, the frugivory species, and in the bottom row we have the plant species that are dispersed by the bird species, and each link corresponds to an interaction between two species. For our work, we use 34 networks, frugivory networks, compiled by Alex Bigot and others last year. From these 34 networks, 33 are quantitative, or this means that they have included also the frequency of dispersal by each species, and 11 are only binaries, just the presence or absence of interactions. And in the total of 546 species of birds that are this way distributed along the molecular phylogeny. So we can see that there's a prevalence of passeriform species, but also there are some other species kind of well distributed along the tree. So this is the network part. We calculated the closeness centrality for now. And on the other hand, we also have the macroevolutionary rates, speciation and extinction, that we obtained by the study by Harvey and others published this year. There's a bug there, but sorry. There are 27 different shifts in diversification throughout the tree. This is the pruned tree containing only the 546 species that we have network data for. And the red circles, they denote the closeness value, like the centrality, the ecological role that each species play in their networks. So having these two types of data, both the macroevolutionary rates and the network centrality metrics, we performed 3 different tests just to check if there is any relationship. So linear models, a mixed model in a Bayesian framework, and a permutation model built on BAM tools. What did we get so far? So at first look, looking at the standardized values of closeness on the x-axis and the standardized net diversification rates on the y-axis, we see that there's not so much correlation between these two data. There's a slight trend, a positive trend, but nothing too exciting. But when we can add some different cofactors, for example, latitude. So when we separate the networks on temperate networks and tropical networks, we see an interesting pattern where tropical networks have a tendency to have a positive correlation, whereas the temperate species seem to have a negative correlation with diversification. And this is maintained when you look at the speciation and extinction rates alone. However, when we performed all the 3 types of tests, only one type of test came out significant, that is between the net diversification and both the ecological role and latitude. But all the other ones are not significant. But it is interesting to note that despite not being significant, this tendency of tropical species showing a positive correlation and the temperate species showing a negative correlation with the closeness is maintained. So there seems to be something going on there. So let's talk about this. What might be going on now? So some possible explanations. The first one, no actual relationship. So it's kind of frustrating, but we have to deal with this possibility. But thinking about more exciting scenarios, one possible problem might be related to the poor estimation, both from the rates, the evolutionary rates, and the centrality metrics. We know, for example, that Harvey, they used a global undersampling fraction for the whole tree to estimate the diversification rates. But this can be improved by using clade-specific undersampling values. So we are running them now with clade-specific values, but it takes a long time because it's a huge tree. But this is something we can do to improve our estimates. On the other side, by the networks, I mentioned that there are 2 different types of interaction networks, and probably calculating the centrality the same way for the 2 types of networks it doesn't make too much sense because the information is slightly different, so we might want to standardize this. possible explanation for this lack of significant correlation is that there are, in fact, more complex scenarios. For example, using only temperate and tropical separation is not enough. When we take a look at all the tropical trees plotted, like the species from each of the tropical trees plotted with the same plot but separated by network, we can see that there is a prevalence of positive correlations, but there are some trees, some networks that show a negative correlation, and this might be adding some noise and weakening the signal that there might be there. And the same thing happens to the temperate networks. And it's also worth noticing that there are some networks that are quite small, so we might have to clean this data so we can have better results. And lastly, we can take a look at different network features, such as modularity, for example, where you have networks that are composed by modules that have a high level of interaction among the modules, but some species connecting two different models. And also nested networks, where you have a highly generalist species and highly specialist species that interact with a subset of the generalist species. So these different characterizations of the whole network can be related to the different behaviors we see in the individual networks. And also, probably these different network features might indicate that those trees, those networks, are in different stages of evolution, like in different assembling moments. So this is something we want to investigate further. Where to go next? So try different estimates of centrality. I showed you only for the closeness centrality. Although they are highly correlated, they mean different, slightly different things, so we want to try to address properly what each of the metrics represents. And also try different cofactors. For example, instead of separating by tropical and temperate, separating by biomes or bird groups or anything like that. And try also to incorporate network properties, not the nodes, the property of the nodes, but a property of the whole network, and like size or modularity or nestedness, to see if this can help us find something interesting. And also, it's a particular interest of mine to have a deep reflection about the tests, because it's already kind of confusing for me that when you estimate diversification rates, is the phylogenetic structure already there or not? Should we correct for phylogeny or not? So this is something, if anyone wants to discuss, I will be very glad to discuss with you. So I just want to thank FAPESP for the funding, the Lab IMEME, my lab, and Luke Harmon for all the help. And this is my tweet handle, and thank you very much.